Hi, so let's now begin part 2 to asynchronous lecture 2 after our little break. As we were talking about, protozoans are often categorized based on what they're using to move. So if we look in this picture here, this is, again, I don't want you to memorize these names, just here's the example, understand the example. Here, these ones use flagella, one long hair-like thing. These use pseudopods, pseudopodia, pseudopodium, that's just like a part of, like a bendable part of the plasma membrane that stretches out and grabs things, and then the rest of the cell will flow into it. Okay. We also have cilia here, one cilia and many cilia, which are kind of like short hairs. All right. So it just kind of gives you a feel for the structures that are used for locomotion in protozoans. So now that we're done with protozoans, let's start fungi. Okay, fungi are eukaryotes. They're in the domain Eukaryota, and they're in their own kingdom. So they have a special kingdom. In general, fungi are bigger than bacterial cells. All right, they're about 10 microns or so. And you can, when you classify them, you can think of them in two big groups. Okay, two broad groups. The first are the yeasts. Think yeasts are unicellular. And they're what we're used often to make wine, beer, bread, stuff like that. And then you have the multicellular. Here's an example of a multicellular microbe, are the molds. They're long chains of cells. So in this drawing here, you have these chains of cells stuck together. They're not unicellular. So these are called molds. And these chains is called a hypha. So this would be a hypha. This would be a hypha, okay, and many are hyphae, all right? And molds are known for being organisms that make a lot of antibiotics. They're also used to make the culinary delight known as tempeh. And I'm going to talk quite a bit about tempeh, not so much because I like to eat it, even though I think it's pretty tasty, but it illustrates a lot of important concepts about fungi, okay? So I'm going to talk about tempeh, which is something you could order at like a vegetarian restaurant, usually. Okay, so when you want to think of unicellular versus multicellular, just quickly think about if you have these growing in a big vat of juice. Okay, the yeasts. This is the yeasts up here. All right, these are the yeasts. They're going to grow in that juice. They're going to grow independently, individually, unicellular. But if you grow in a big vat of juice, the molds here, they're going to grow like a mat of hyphae, okay? I know that sometimes you think, well, you go in the lab and you streak out some yeasts and they'll grow as a colony. And you'll say, well, Jeff, look, they're all stuck together. But that's kind of an unnatural thing. When we talk about yeast being unicellular, molds being multicellular, we're thinking in nature, not in a lab, okay? Now let's get to tempeh. So tempeh is something you can go eat. Here's tempeh, all right? And what it is basically you have soybeans here, so you can see the beans. All right, here's your beans. And what's holding them together are the hyphae of the mold, okay? So here you can see kind of furry-like hyphae, and they're actually going through and around and sticking to and going through the beans. And so like the beans are sewn together. So for a simple hokey, what I would say, if I just had like a necklace of pearls, and you'd say, okay, you've got these pearls, what's holding them together? Will there be some kind of string or something going through each pearl? Well, just think of the hypha or hyphae of the mold interconnecting one, going through one soybean into the other. All righty, that's how it's held together. Now, this food goes back many hundreds of years, and it actually was very important in saving the lives of prisoners of war. American prisoners of war being held in Japanese prison camps. And you're like, well, how can that, what has that got to do? It doesn't make, well, just stick around. I'll explain it. So, going back to just general fungi are not photosynthetic. Okay, they can't make their own sugars using sunlight. Basically, they are heterotrophic. They eat whatever they're in. whatever they happen to land on. Okay? And it's important to realize that they are not movers. Fungi are not movers. They don't have flagella or cilia or pseudopods or anything like that. So if they can't move to go to the food, they basically have to hope the food gets to them. They land on the food, and they have to be able to break down that food that they land on. In order to do that, they use enzymes. All right? So they are going to secrete. They're going to, going back to earlier in the lecture, they're going to secrete proteins called enzymes. All right? The enzymes are going to have to go from the, here's a little review, made in the ER, go to the Golgi, and then the distribution decision says, okay, let's send them through some vesicles, and then those vesicles hold the enzymes. So here's the enzymes in these vesicles. Then they're going to go, those vesicles are going to fuse the plasma membrane, and here they can come out. And now, once those enzymes are out there, they can break down whatever food they're on. All right. So I always think of fungi like my brother-in-law Tony. He's a very sweet guy, but he's not the most active guy. And this is a story, a true story. My sister, his wife, came up to him at a banquet and she said, well, why, you're sitting here at the table, why aren't you eating yet? And he goes, because you haven't gone and got me any food yet. Tony has a lot of other good points. But I always say, Tony, you're like a fungus. You're a lazy couch potato. You just sit there and expect the food to come to you. All right? And that's why I think of that. Okay. Now, if you're a mold or a yeast, a type of fungus, what must you need a lot of to produce these enzymes? Well, you must need, obviously, just like I told you, you need to have a lot of ER and Golgi, because your livelihood, your survival, depends on your ability to send enzymes from the ER to the Golgi, through these secretory vesicles and out of the cell. So they got a lot. They have yeasts and molds have a very advanced secretion mechanism. Okay. And this, going back to my little teaser from the previous, this is why tempeh is so nutritionally rich. So tempeh, again, is just soybeans that are being broken down by the enzymes of the mold that's growing through them. The mold, by the way, a little cocktail party comment, it's called Rhizopus. That's the genus. But that's not a big deal. Okay. So let's thinking here, how about the POWs? Explain that story. Well, the POWs, prisoners of war, are getting very little food in the camps. All right? Very, very little food. And here's the thing. When you eat raw soybeans, they go to the human gut. They're very complex structured. They're very hard for your body to break down. Your body doesn't make sufficient, really, enzymes to break them down efficiently. All right? The stuff in the soy, the mac needs to be broken down pretty well for the body to absorb the nutrients. Okay? Now, if you're eating lots and lots of food, if you don't get all the nutrients from your soy, it's not a big deal because you're eating a lot. You can get it from other things. But the prisoners of war, they were pretty much being almost starved to death. They were getting very little food. So they had to be able to absorb all the nutrients that were in the little soy they were getting. All right? Okay, so they had to be able to use all the nutrients that were in the soy they were getting to survive. So the soy, once it was in the tempeh and the mold was secreting enzymes, it was being completely broken down by these highly sophisticated enzymes made by the mold. So now, because everything was being broken down, the nutrients became usable. Okay, I know that's not the greatest word, but that's the easy one. These nutrients could be used by the human body and absorbed by the human body. All right? They became used. They were digested, broken down. They became usable. All right? That's why they got the accessible from the tempeh, and they were able to survive. Versus those POWs that just got raw soy, they basically starved to death. So the mold in the tempeh saved the POWs. That is a true story. Last thing on fungi, here you have your yeasts. Up here, again, unicellular individuals. Here you can, under a microscope, you see the mold. What we're looking at here are just long individual hypha, many hyphae together, long, thin, they're interconnected. There's multiple cells here. How about in this picture? Where do you see them? Well, you can see the mold growing on this wall. This is from a flooded area from Hurricane Katrina. There's the mold. It's growing on the wall. And the yeast, well, they at least were down here in these bottles, and they were used to make alcohol that was in these bottles. So, when people were living in these flooded out, really bad areas, the molds were growing in there, they're depressed, and they resorted to the product of another fungus. Okay. Our next one is a unicellular algae. All right, so we're going to unicellular. So they're eukaryotes. So just reminding, what does that mean? Nuclei and other kinds of organelles, right? Just a reminder there. They're a member of the protist kingdom. All right, it's a little different kingdom than the fungal kingdom. So they're all the domain eukaryotes with the fungi, but they're a different kingdom than the fungi. Kind of important to remember that. They're in the kingdom, again, with the protozoans. These are about 10 to 20 microns, usually in diameter. But, of course, there's a few that can get really big, like 200. They're bigger. So these are bigger than bacteria, most yeasts, things like that. And they're referred to they're plant-like organisms because they do photosynthesis. But they're not plants because plants are multicellular. So we say if they're unicellular, then they're going to be in the protist kingdom. Okay? They're unicellular, they're in the protist kingdom. If they're multicellular, they're going to be in the plant kingdom. So that's how they make that differentiation. So think unicellular algae, protists, eukaryotes. What's key about them to know? Well, they have cell walls with cellulose. They're eukaryotes, so they can have a chloroplast that contains the chlorophyll. And they have your usual organelles that you think of a eukaryote. They have the nucleus, mitochondria, lysosome / vacuole, so forth. These are some examples of some unicellular algae. I'm going to go into a little bit about them because this is pretty much the only time I talk about them in this whole course. And they're really cool. There's diatoms. All right. Notice it almost looks like they have a skeleton. Doesn't it look like they have a skeleton here? Well, I'll talk about what that skeleton-like thing is. And here's your dinoflagellates. All right, they have two flagella, which don't show up here. But you can see the green, the chloroplasts in here, all right? They're holding the chlorophyll. These are pretty cool. Well, I just want to talk a little bit about them, okay? So, first of all, these are important because they're in the food chains of the ocean. They're like the producers. They're the ones that convert the light energy of sunlight through photosynthesis into chemical energy of sugars and other macromolecules. They're the beginning, all right? There gets what eaten by some heterotroph, which is eaten by a bigger heterotroph, and so forth. So they're really important for the ocean food chain. Specifically diatoms, what's kind of interesting, maybe what's something, I always like to talk about money. So in terms of financial, why there's a lot of interest in diatoms? Because in their cell walls, okay, all right, that you have cellulose, absolutely. But you also have, in diatoms, you have silicon dioxide. And think of something like sand, silicon dioxide, hard, rough things. And this is what you might buy diatomaceous earth. It's, again, it's thin, skinny, like sand-like stuff because they're basically dead, skeletonized diatoms. So what you have here is the outside, the cell walls of dead diatoms. And they call it diatomaceous earth. And it's real fine and rough, so it can be used as a filter. And it's often used in cleansers. So a lot of cleansers, they're rough and scrubby. Why? Because they have old dead diatoms, diatomaceous earth in them. And you're basically using the silicon dioxide in the cell walls to do the abrasiveness, do the cleaning. So that's a little bit about diatoms, all right? They're pretty interesting. They're non-modal, okay? They're non-modal. They usually don't have any flagella or anything like that. The second group of unicellular algae I'm going to talk about are the dinoflagellates, which I showed you. And what's interesting about them is not a money thing. but it's the fact that they're involved in what's called periodic red tides in oceans. Now, what you're looking at here is coast, and you see this redness here. And this is where you see an overgrowth of dinoflagellates. So they say, oh, these areas are red tide, all right, because there's an overgrowth of dinoflagellates, and they turn things red. And you're like, well, I mean, come on, can they really hurt you? I mean, they have two flagella for movement, dinoflagellates. They are protists. They do photosynthesize. But the problem is they make toxins. When they overgrow like this, and there's a lot of them, they make toxins. Example is saxitoxins. And what happens is this, that the dinoflagellates end up being filtered and get concentrated inside of like certain seafood, like filter feeds, like clams and stuff like that. And so they get concentrated there. So if the dinoflagellates gets concentrated in these shellfish, so are these toxins. So if you end up eating this stuff, all right, these shellfish, like clams, okay, these ones that came from a red tide area, you could be eating a lot of these saxitoxins. And these saxitoxins, when they get in your body, what do you think they lead to? Well, the key thing is, what's true of all of them is they lead to paralysis. Paralysis. Oh, geez, that's a long word. Paralysis. They'll literally paralyze you. So there's some people, they'll eat them and they'll start to lose feeling. Can you imagine that? They start to lose feeling in their tongue and mouth and things like that. And that's mild, but it can be really bad. That it can cause paralysis throughout the body. And there are some examples of people getting really, really sick and even dying. So that's why you should never buy fresh seafood that came from a red tide area, even though it might be really cheap. There's a reason it's really cheap. Okay? Danger. Not worth it. Okay. All right, let's go to the next group. That's pretty much all the time we're going to talk about unicellular algae in this whole class. Next group is, we'll talk about these a lot later, but talk about viruses. And viruses are neither prokaryotic or eukaryotic. They're living. Why? Because basically they say a living thing is something that can reproduce kind of by itself. All right? It has that independence of reproduction. Viruses, they really can't reproduce unless they're inside a host cell. So outside a host cell, they're dead. They don't reproduce. So that's kind of their definition. So that's why a lot of people don't like to call them living things. We are, because this is a microbiology class and they're such an important part. So we're going to consider them living organisms, okay? But they're neither prokaryotic or eukaryotic. They don't fall into either group because they don't have the basics of ribosomes, plasma membranes, cytoplasm. They don't have that. So they're neither prokaryotes or eukaryotes. All right? They have some pretty cool, here's just some models of what they look like, okay? All righty. Here's a lot of different ones. And you can probably heard of Hep C. I'm sure you've heard of coronavirus. That's one of these is what causes COVID-19. Probably heard of smallpox, flus, influenza, okay, herpes, all right, viruses. Generally size, they're smaller than most bacteria. In general, most of them are 0.02 to 0.5 microns. That's 20 to 500 nanometers in size. Okay. They can, again, only replicate inside a living cell. And we say the living cell is a host. Outside the living cell, it can't replicate. It's dormant. Okay. And depending on the virus, it can last outside the living cell maybe minutes to days. All right. Meaning that, okay, some viruses, they get outside a human cell, 5 minutes, they're dead. You take them, you put them back, they can't infect the human cell, they're done. Other viruses are really hardy, meaning they come out of a cell, they can last on the surface of a desk, maybe the table you're sitting at right now, and they can last for days. Then they get next to a living cell, and they can go into that living cell and infect and reproduce. So that's what it's meant by kind of like how hardy they are, how long can they last outside a human cell and still be effective. So there's our examples. We kind of went through those already. And I'll spend a little time now. I'm not going to spend a lot of time on COVID-19 until later, because really you don't have enough stuff to understand and appreciate it. Okay, later we're going to go in all about viruses, viral life cycles, and things like that. And then you'll be in a position to really understand and discuss COVID-19. But we got to know a lot of stuff before we get to that. So please be patient. So what's true of viruses? Viruses are very simple. They contain some kind of genome, DNA or RNA. So here's a picture. You got DNA or RNA. You got a capsid. And a capsid is just kind of like a structure, a protective structure. Here's your capsid here. It looks like a kind of hard, kind of like a weird little structure here. It's got sides to it and everything like that. And you got your genome, and you got some soluble proteins in here, inside the capsid. There's no organelles. Notice there's no organelles. There's no cytoplasm. There's no ribosomes. Very simple, not a prokaryote. Now, what might happen is they might have a membranous-like structure outside the capsid, and that's known as the envelope. Here they call it a bubble of fat, but that's not really the best thing. All right? And sometimes in the envelope they have these proteins, okay, which we'll talk about another time. And viruses just use and abuse. Okay, so here's a picture, electron micrograph of some viruses. It's not a model, it's an actual picture. And you can these have an envelope with some proteins on the surface. And you can imagine inside, you can't see it, but you got your capsid. And you're going to have inside your capsid, you have a genome, and you have some soluble proteins. And basically, what are they living? They're basically waiting to go inside a host cell, like a human cell, take over everything. And basically, all of the parts of the virus are going to be replicated. So the capsid parts will go in and they'll be replicated. Many, many, many copies of those parts. The genomes will go in. They will be replicated. Many, 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 many. And then all those parts are going to come together like an assembly line in a car plant. And finally the cell, because it's been working to death to make virus and not take care of itself, is going to die. Okay. Use and abuse. That's what viruses do. Just in very simple terms. Let's look at the next one. This kind of goes into this a little bit more. So I don't want you to memorize these steps. Okay. I'm just understand what I wrote on the previous slide. You have a virus. In this case, it's a flu virus. It enters. This all I want you to know. It enters. It basically releases all its parts. Okay, it releases all its parts. So all its parts are there. And then basically the cell is taken over by the virus. And the virus basically commands the cell, make many copies of my genome. Make many copies of my capsid proteins. Make many copies of my soluble proteins. So all these copies are made. All right? And then they come together, kind of think of like an auto plant assembly line. All those parts come together to make new baby viruses. So in this case, all right, we've got three new baby viruses. And then those 3 baby viruses are released by this cell in this picture, and the cell dies. All right, and that happens with the flu in your body. That's what happens to your cells. Cold, you have a cold virus, that happens. And even SARS-CoV-2. So SARS-CoV-2 is the fancy name for the COVID-19 virus. Okay. Now what's really interesting is this. In this picture, which is not accurate, we have a flu virus, one coming in, taking over, copies are made of all the parts come together like an assembly line, and 3 babies are made. Okay, so that's a 1 to 3 relationship. What do you think it is for SARS-CoV? Let's imagine SARS-CoV virus, one of them enters one of your respiratory cells. How many potentially can be made, produced in that one cell? How many new babies can leave? 5, 10, 20. Try 1,000. 1,000. So theoretically, one baby SARS-CoV-2 virus enters a respiratory cell, takes over, all the parts are reproduced, the parts come together, 1,000 new ones come out. All right, think about that. That's why you get sick pretty fast, right? Because one virus goes into one cell, 1,000 come out. 1,000 new ones can attack 1,000 cells in your body. Okay, 1,000 times 1,000, 1,000,000. Now you have 1,000,000 viruses in your body. They attack, each one attacks a cell, 1,000,000 cells. So you can see that 1,000 times 1,000 for every generation, potentially. Scary, huh? All right, so enough about viruses. So what's going on in this picture? This is a nice kind of a summary picture. What's going on in this picture? This takes a little bit of everything we've talked about today. So I want you to look at it and kind of like piece together what everything means. First of all, you have Rickettsia. Then you have lysosomes. Okay. You got mitochondria. Don't worry about glycogen. Okay, that's not a big deal. And you got phagosomes. And phagosomes are lysosomes that have stuff in them. Okay? So what we're saying is you got the Rickettsia are inside lysosomes, inside this big cell. So think about that. Rickettsia inside lysosomes, inside a big cell that also has mitochondria. So first of all, what are Rickettsia? Think about this. What are Rickettsia? They're a type of small bacterium. Where are these Rickettsia? They must be inside the lysosomes of a big eukaryotic cell, right? So what we have here now is we have Rickettsia. They're inside the lysosomes. So what's happening? Well, they're in there. Probably the lysosome is trying to break down these Rickettsia, right? These small bacteria that could hurt the cell. Well, the body cell, a eukaryotic cell, because it has lysosomes and mitochondria, is basically trying to kill the Rickettsia. So that's kind of what's going on here in this picture. All right, if you know all the basics into what each is doing, you can kind of put together and figure out what's going on overall. Hope that makes sense. All righty, so now for the last 25 minutes of lecture, hopefully not that long. We're going to talk a little bit about taxonomy. And I know taxonomy is just classifying. We're talking about microbes into groups. All right, so let's kind of talk, and we're going to see the importance of it. There is a usefulness to it. All right, so and here we're going to start off by looking at this question, because you've already been doing a little taxonomy now, and we're going to get further. So you got Rob U. Blind. I love these names. Sorry, I make them up. This is what I do for fun. He's a microscope salesman, and he's showing you samples of Hensenula polymorpha, a yeast, and Endoscrinium campanella, a dinoflagellate. What's true of both cells? All right, so this is a classic question I like to have on tests, where I say, here's two cells. Okay, what's true of both of them? The first thing you want to do is ask, is each cell a eukaryote or a prokaryote? I'll tell you right now, that's the first thing you do. So you go, okay, it's a yeast. Okay, that's euc, big E for eukaryote. Dinoflagellate, it's also a euc, big E for eukaryote. No, so there's a lot of similarities, but then you got to think, okay, from what we've been talking about, what's true of yeast, fungi, what's kind of unique to them, what's true of dinoflagellate, what's true of them. So you should think about that to begin with. Okay, what are some differences between yeasts? What are differences of dinoflagellates? They're both eukaryotes, so what's true of all eukaryotes? Okay, then look at the question. Number one, they have 60S ribosomal subunits. Well, both of those eukaryotes. Do you find 60S ribosomal subunits in eukaryotes? 60 plus 40 is 80. So yes, that's a good one. Yes, true. They contain flagella and ER. They do contain ER because they're both eukaryotes. But do both contain flagella? Well, remember yeasts, because my brother-in-law Tony, lazy couch potatoes, no structures for locomotion. Nope. That can't be right. Nope. They're smaller than 2,000 nanometers. 2,000 nanometers is 2 microns. These are in the neighborhood of at least 10. Always be able to do the conversions, okay? They contain chloroplasts. Um, Dinoflagellates do, but yeast don't. No. Yeast don't photosynthesize. So what's the answer? The answer is A. A is your answer. Okay. So let's now really get into more what people think of as taxonomy, okay? But like I said, we've been doing already. Taxonomy is just classifying. It's arranging related organisms into categories based on their degrees of similarity. Alrighty. So, and you've seen these taxa before, all right? You have things, things can be in the same domain, but that doesn't mean they're really similar. If things are, okay, the next thing, more specific, things are in the same kingdom, there may be, all right, they're a little more similar. They're in the same phylum, a little more similar. Same class, yeah, a little more similar, but let's say they're different orders. Then there's some pretty big differences. So they share some things, but they're different. But let's say they're the same order, the same family, the same genus, and the same species. Now you're saying, well, wow, they got to be pretty close. Okay? It's also important to remember that, if something's in the same species, if you have two things in the same species, then they must be in the same genus, same family, same order, all the way up. So don't forget that, okay? So basically, if they're the same, whatever it is, everything above it, the larger groups, they're always in the same member of. So two things can be in the same genus. They must be in the same family, but they don't have to be in the same species, right? Okay, so just kind of remember that. This is kind of Bio 51 stuff. And it's really important to know, if you have a microbe, what taxon it's in, all right? It helps identify it. This is a little short YouTube video where it's from England, and it's a public health lab, and they're trying to determine if a bacterium that's kind of an unknown that's been involved in some food poisoning outbreak, is it Salmonella? And Salmonella is a genus. And basically, they're in this little video, they're talking, this lady with a wonderful accent. I mean, she's talking about some of the tests they do to determine if it is in the genus Salmonella. All right. And so the tests, we're going to talk about those kind of tests, those things that are used to determine what taxon, determine the classification, the taxonomy of an unknown. All right. I can't play this video. This is one of the problems with the Canvas player program I'm using right now, where I'm recording this. I can't play videos in there, but you can take that link, put it in your browser and play it. It's only a minute and a half. And like I said, it kind of illustrates some of the things I'm going to talk about next. And again, she's got a great accent. So how do they do classification of microbes? And we're going to mainly focus on eubacteria. What do they base it on? What kind of tests? What kind of things? Number one, physical characteristics. This goes back for years and years. They look at the shape of the mystery microbe, its size, its arrangement, how it clusters, what pH and temperature requirements for growth. That helps them identify things, classify it. Secondly, there is biochemical tests. And they've been using these for decades now, pretty old style. And it's kind of based on a micro 20 questions. And these tests are always give yes or no answers. Okay, they're not open-ended, they're yes or no. So it's like if we were playing 20 questions with a famous basketball player, and I said, I'm thinking of a famous basketball player. It all had to be yes or no. So you could only ask yes or no questions. You couldn't say, what team does he play on? You'd have to say, does he play on the Lakers? Yes or no. Does he play on the Warriors? Yes or no. Is he a forward? Yes or no. Does he score 20 points or more a game usually? Things like that. Okay? So for microbes, the questions are like, does the microbe make a certain compound when it's grown on the sugar? Yes or no. Does it make another one? Does it make this compound when it's grown on a lipid? All right? Yes or no. And basically, when you have enough yeses and no answers, you can figure out what the microbe is. All right? Just like you could by playing 20 questions about a basketball player. So what are they looking at? In these biochemical tests, you say, well, okay, how does the microbe, what does it make when it, does it make this when it's fermenting carbohydrates? does it produce certain products on certain substrates? And probably the most famous kind of biochemical tests are these entrotubes or entropleury tests. And I'm going to show you one in the next slide. And it's just a bunch of biochemical tests that are used for identification. When you do the Labster lab in a couple weeks, they're going to talk about an identification. They're going to talk about some examples of biochemical tests too. So I'm just giving you a little preview. You'll get a lot more details soon. So here is your classic entrotube, entropleury test. And again, I don't want you to memorize that. Just understand the concept. It's pretty simple. So in this thing, you've got about 12 different compartments. And they have, so here we go, 1, 2, 3, count them. And they have like different compounds, things for the microbe to grow on. So what you take is you basically take the microbe and you put a little of it in each of these chambers. So some chambers, like this chamber has lactose. This chamber has arabinose. All right, this chamber has sorbitol. And you wait for the microbe to grow and see if it makes something. All right, for instance, does it break down sorbitol? Does it break down arabinose? Does it break down lactose? Does it grow here and produce hydrogen sulfide? So yes or no. All right. Does it have urease? Urease is something that breaks down urea. And if you look here, when you don't see a color change, okay, let's say lactose, arabinose, you'd say, oh, these guys here? No, nothing's changed here. Nothing's changed here. They're all no answers. But let's say you look at urease. You'd say, oh, wow, yeah, that's changed. This has a positive yes for urease. So I know it's urease positive. I get H2S. Oh, look, it made H2S. That's a yes, it made H2S. So don't worry about all this stuff here. But you don't need to understand how the circling and everything goes. You don't have to worry about that, okay? The principle is that based on the yeses and the nos, this profile helps you figure out what the microbe is. Okay, that's kind of how it works, right? That's how biochemical tests work. And you'll learn a lot more when you do the Labster lab. Okay. So what's the third way? Third way is using serological tests. And serological tests, when you see you want to think antibody looking for antigen. So antibodies, you should have heard these before in earlier bio classes, but just give you the basics. Antibodies are proteins that bind to a specific target antigen. Okay? Antibodies bind antigen. So here's an antibody. All right? And if it's very specific, it's only going to bind a specific antigen. So in this case, it's binding the antibody is binding the antigen. So you'd say, oh yes, it's a positive reaction. It's found something that it fits. So let's say this is the anti-protein A antibody. Okay, and it's looking for protein A. And let's say this is protein A. And you say, oh yeah, the anti-protein A antibody's there. It finds protein A, it binds. And so we can say, oh, this mystery microbe, it's been bound by anti-protein A antibody. It must have protein A as its antigen. And so I now know, okay, that's one clue, maybe not definitive, but it's a clue to say, oh, this mystery microbe has protein A on it. And based on that and some other things, I can figure out what it is. Because if it contains protein A, then it might be whatever. So that's kind of how serological tests work. You work based on antibodies binding antigens if they're present. Okay, sounds good. The next one is nucleic acid analysis. And they call it molecular taxonomy. Because usually what they're doing is they're looking at the rRNA gene sequence, okay, in a mystery microbe. Okay, so it's pretty easy just to PCR up certain rRNA genes from a mystery organism. Okay, and these are used because you can have two different but closely related microbes And they might have the same gene sequence for some enzyme, hexokinase. So they're different, but the gene sequence for their hexokinases are the same, even though they're different microbes. But usually the rRNA gene sequence is very particular. And you can have two really close related microbes, but if they're different microbes, their rRNA gene sequences will be different. So rRNA gene sequences are usually pretty unique to the microbe, to the species of microbe, let's say. Okay, so what happens is, and you're going to do it's going to be a lab. We got it from the HHMI. In a couple weeks, you'll see it. It's a little video. You'll do a little simulated lab, and basically they're going to say, okay, this is the rRNA gene sequence of the unknown. There it is, and then you basically run a program, and you run it against all the rRNA gene sequences of known microbes, and the program looks for a match. And because these things are so different, depending on the microbe, you can say, this has got to be, it's a perfect match. Well, it's got to be this microbe. All right. So you kind of get that. So that's how you're looking at the rRNA gene sequence. Here's just a real cool example. You're looking at 4 microbes here, rural, and they gave these names L11, 6, blah, 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 and they lined up the rRNA gene sequences, okay, of each of these microbes. So they PCR'd them up, they sequenced them, and they're comparing them. These 4 different organisms right here, 4 of them, and they're just comparing the rRNA gene sequence. And just as a reminder, rRNA genes encode ribosomal RNA that gives you this really cool structure that make up ribosomes, all right? So it's the rRNA itself, not protein, RNA itself. Once it's made, the rRNA version is made, it folds in these shapes, and those give structure to the ribosomes. Now, if you compare their DNA to begin with, what do you see here? Wow, they're almost identical. But look, hold it. Looky here. There's a difference here, okay? These guys, so this one's missing, this G, and you got a CG instead of a CC. So you'd say the bottom 3 are identical. Maybe they're all members of the same taxon. Maybe they're the same species. But look at this, rural. Rural has a difference in the RNA gene sequence. So you'd say, rural is not in the same taxon, maybe species. It's not in the same species as these other 3. These 3 are in the same species. But rural, because of the difference in the RNA gene sequence, is in a different species. That's how we're using it for classification. All right, cool.
And the last thing, we'll talk about doing taxonomy with proteomics. And we'll use the example of a very famous, handsome, well-known professor in our department, Dr. Vieira. All righty. So what they do when they do a proteomic analysis means they're not looking at one protein. They're looking at a population of proteins. That's the idea, looking at a population of proteins. So what they basically do is the people who do proteomics like Dr. Vieira, they'll basically take the proteins from a known organism. So something that's known, a known organism, okay? And they'll take those proteins and they'll get the sequence of those proteins. So they'll sequence them. And they'll get, therefore, kind of like a profile. So they'll know, okay, we'll know all the proteins and the sequences of the proteins of that known microbe. Okay? And they use a machine to do this called a mass spectrometer. So this is called mass spectrometry. All right. So basically, you now know the proteins, the sequences of proteins, a lot of different proteins in a known microbe. Then you take an unknown microbe, okay, and you basically take its proteins, you chop them up, you get the sequence of them, okay, you're going to get the sequence of them now, all right, and you're going to compare those proteins of this unknown microbe to the proteins of a known microbe, and you're going to look for a match. And that's kind of how you can figure out, okay, what's my microbe? What is it? Or at least, if it's totally new and novel and original, what's it most similar to? So you'd say, oh, the proteins of my unknown are most similar to the proteins you find in E. coli. So it's got to be very similar to the bacterium E. coli. So I can kind of place it in that, near that taxon. Okay. All right. So that's kind of the last of the 5 tools that are used to do taxonomy, to do classification. Well, thank you so much for listening to me today. Take care and stay healthy. And hopefully you'll listen to this before you go to the live lecture. Okay. Bye-bye.